All right, good morning, everyone. So this morning I'll be covering some disease management in soybeans and also throw just a little bit of some small grain highlights in at the end. We'll begin today talking with soybeans. I'll share some of our full season foliar fungicide efficacy trial results, begin to go into some surveys and summarizing some of the work we've been doing from 2019 to 2021. We'll talk a little bit on soybean cyst nematode seed treatment trials, and then at the end, wrap up with a little bit on fusarium head blight, both looking at some wheat timing trials and doing some fungicide screening. So this first slide I have up here today is from our foliar fungicide trial for 2021. It was a very calm year as far as foliar pathogens go for soybeans. We didn't have any ratable disease within this plot. So the metrics that we ended up taking, we looked at green stem a couple weeks out from harvest. Then at the time of harvest, we also looked at average purple seed stain and Diaporthy phomopsis seed decay. So we're looking here to see if any of these R3 or R5 timings have any impact on that final soybean seed quality. For purple seed stain, we did get a few differences that came out. Our control seeds were kind of ranging in that 36 to 46%. And this was the number of seeds within a 50-gram sample. And then you can see that we had a fair bit of purple seed stain out in this field this year. This one was a little bit later of a harvest date. And then we had some of that Diaporthy and seed decay out there as well. Taking a look at the yield, we did get a little bit of separation that came out with our controls in that 46 to 50 range. And we had a few treatments where we saw a 3 to 4 bushel increase. And some of that was tied with those seed quality ratings. Where I'll spend most of the time talking about today is on some of our work with soybean cyst nematode and nematodes in general for soybean management. And we talk of this because for the past many years, SCN consistently ranks as the most damaging soybean pathogen across North America. The reason for this is it can persist in the soil. It's kind of, once you get it, it's sort of stuck there, and it can make many generations per year. So we're going to have these females that eventually will fill with eggs, and that body turns into a cyst that can survive in the soil for long periods of time. It's kind of depressing how long these can survive. There's studies where folks are looking at longevity in 10 plus years they can stay. And then once these hatch out, those eggs will be these J2s that are going to swim looking for some root exudates and feed that, make a feeding site with the root. We can then have as many as 3 to 6 generations per year, depending on what kind of environmental conditions we're having. So really an opportunity to get a lot of nematodes in the soil, and because they're underground and happening, often we don't know this is happening. So SCN is often dubbed the silent yield robber, and you don't really know it's there until you start looking for it. And especially if you have a case where it's across most of the field, you may have stunting, reduced yield that you don't even realize is happening. You might just think it's from just the way that field is. And we always run the kind of added piece here where we have a lot of sandy soils, and often those sandy soils are really favorable for nematodes. So it's hard to know, is my soil type driving it, or maybe there's these nematodes that are there as well. I have this picture up in the corner to show that resistance within the soybean plant is a great way of controlling this. And for many, many years, we had pretty excellent resistance that came from the PI88788 gene. And so with that, you can see on the left versus the right, the only difference here is just that we have that resistance in the plant. Unfortunately, we've used this resistance gene for many decades now, and what we've seen is a slip. So when it first came on the market, we would only really get about 10% of reproduction or less. We're now at the point where on a lot of our local lines here, it's 65, 68, 70% reproduction. So we're still getting, maybe that 20% of control, but we used to get 95 or more % control. So over time, this is allowing those nematode populations to creep up every year, and we're just not getting that control we once did. So There are a few new resistance genes on the market, but at the moment, our options are still pretty limited. The PI88788 still in, 95 to 98% of soybeans that are on the market. There are a few beans out there that have a Peking resistance gene. And there is a new one out right now that's still only in low maturity groups like 2-3s, but we're hopeful that maybe those will expand in coming years. So to begin with some of this work, we were interested in just getting a handle on nematode distribution within the area. And we were very interested in soybean cyst, but we also wanted to learn about root knot nematode and potentially if there were any other nematode types that are out there that we needed to be concerned about as well. So a grad student in my lab, Lexi Kessler, has been involved with this project for many years. And we've had a variety of sources that are supporting us through both the Delaware and Maryland soybean boards, the Atlantic Soybean Council, and our work with the Soybean Cyst Nematode Coalition. When you go out to do nematode sampling, there's kind of two approaches you can take. So one can be predictive. So if you're just interested in knowing, does this field have nematodes? I don't know if there's nematodes out here. And so for that one, you want to take kind of a zigzag M or W shape pattern and just work your way through the field. With nematode sampling in soybeans, we typically target our sampling in that August, September timeframe. So we're catching it at the end of the season where those nematode populations are kind of at their peak. And again, we're targeting this time frame because with soybean cyst and root knot, they're actually attaching to that root system. So they're forming, on that root system, they're going to be there so we can actually capture them. When we do some of our corn sampling, it's a little bit of a different approach because some of those nematodes are actually migratory, where they'll feed early and then move deep within that soil profile. The other type of sampling we can do is diagnostic. So this would be, say you have a spot in the field where just something is not right in that spot and you want to know what's going on. Then our approach here is to typically sample within that problem area, but then we also want to take a sample from a healthy area so we can use that as a point of comparison to see if nematode number is what's driving those differences. So over the past 3 years, we've done quite a bit of sampling across Delaware and Maryland. And I have just a kind of a summary slide here of showing the percent of recovery that we got in those samples. And just because nematodes are there, it doesn't always mean they're a problem. So we do have these economic damage thresholds. This is something we're in the process of reevaluating. These thresholds can be really difficult to try to come across. And a lot of these are a combination of things that we've used from North Carolina, Virginia, the Midwest, but we're trying to hone in on these. And based on our current thresholds, you can see that we do have a variety of nematodes that we've been finding, and quite a few that are actually above those population thresholds. So kind of as we were expecting, soybean cyst was very prominent. It was in almost 57% of samples. And within that 57%, about 66% of those were at that high threshold. So that means, we're expecting economic impact. There's going to be some yield left on the table because of those. We also saw quite a bit of root knot nematode, and 36% of those samples were above threshold. Other nematodes that were around that we found quite a bit was lesion and lance, as well as stubby root and stunt. And then you can see another one here, spiral, that is often really abundant, but that is one that's usually thought to have to be really, really high before we ever see impacts of those. So this is a project, like I said, that we're currently in the process of summarizing. And so there'll be a publication and some fact sheets coming soon. So we'll be sharing those within the weekly crop update as we get these finalized. But a little snapshot there that we certainly have plenty of nematodes in the area. And so next our questions go into, all right, we've got nematodes. What are some of the approaches that we can do to manage? Resistance is that the one that's the best, but we don't have as many options as we used to, as we've seen this decline of PI88788. So what are some other approaches we can take? One of those is looking at nematode seed treatments. And the two probably most common on the market right now are Olivo and Saltro. Both of these are actually fungicides that have nematocidal activity. So we can see activity on SCN, root knot, reniform lance, and lesion on both of these labels. But because these are fungicides, the other big target here is that they're actually targeting Fusarium virguliforme, which is the causal agent of soybean sudden death, or SDS. And SDS is something we have kind of spotty here. We didn't really see much at all in 2019, 2020, but this year in 2021, there was quite a bit of SDS that I got called out to take a look at. We also, it seemed, there was a trend to earlier planting this year. And so the earlier that you plant, the infection with that SDS is going to happen in those early days, right when the soybean hits the ground. So when that bean's in the ground longer, colder soils, all those things are going to favor a higher chance of having Fusarium virguliforme be able to infect. But then we don't see the symptoms until the plant goes reproductive, and we see that stress late in the season with those really well-defined kind of bright veins and some of the leaves. But I will note that soybeans only have so many ways of telling us they have a problem. So just because you see a bright leaf out there doesn't necessarily mean it's SDS. There are a few other pathogens that we want to think about as well, but I'll save that talk for another day. I do want to note that because of these, both the fluopyrim and pitiflumetafin, they do have some early season Septoria brown spot control listed on the label too. And this is something that I've observed in our seed treatment trials. So especially when you're planting early, the value of these seed treatments really seems to come from helping to get that seed up out of the ground. You can see here, this is 18 days after planting. We've got our plain seed over on the left, Olivo in the middle, and Saltro on the right. And this was from some April planted beans. 28 days later, you can see that these plain seeds are still trying to crack out of the ground. And while our other treatments are pretty much out and happy. And this particular year, when we planted in April, this was really put these beans through the ringer because they got there was some crusting, there was a frost that happened late that year, that May 10th frost. So they really were challenged. We took soybean cyst nematode soil samples. We looked at these both at the time of planting, and then we looked at the end of season, and we also took some mid-season ratings as well, where we took a soil sample, but we also then dug a sample of roots and counted those live cysts that were on the roots. And so in our 2020 trial, we were seeing some reductions at that final count for the live cysts. You can see in the yellow bar where we were looking for the live cyst, our Saltro live cyst number was a little bit lower that year. Interestingly, our initial populations were very high in 2020. And so this is something where I need a few more years of data to kind of begin to catch these trends, but we do always kind of associate a cold winter helps to knock things back. And this was a pretty mild winter. And so I'll show you the 2021 shortly, where we had a little bit lower populations coming into it. So with that plain seed, the Olivo and the Saltro, we were seeing, as I showed in those pictures, differences in emergence rate, where at that very first rating, our Saltro had emerged the most, Olivo was in the middle, and our plain seed was the slowest. By 28 days, the Olivo and Saltro were right there together, and we still had that delay in our plain seed. To capture canopy greenness, I'll use this rating called Canopeo. And while this wasn't significantly different from the eye, visually you could see differences where that plain seed lost its leaves a little sooner this year. And a lot of this, I think, had to do with that low canopy Septoria brown spot control that we saw from some of the fungicide activity within the Olivo and Saltro products. When it came to yield, these didn't come out significant, but we numerically had, a 2-3 bushel increase, which, as I'll kind of show throughout the day, is pretty normal with seed treatments. When we're putting this seed treatment on, we're going to have that little bit of protection early on, help get that plant established. but that product's going to wear off eventually. So as we learn with a lot of nematode things, there's not usually that silver bullet. It's sort of just things that we can add that try to chip away at the nematode problem and try to slowly add a little bit of yield back in. We repeated this trial in 2021. So in these bars, you can again see those soil samples that we took in blue at planting, 30 days in the orange, and then gray was our end of season. And then we had those live cyst counts again that around 30 days after. So we do these live cysts about 30 days in. That's when they're really visible on the root system. We can extract them really easy and get some good counts. And so this number is trying to capture that early season activity. You know, by the end of the season, we've had multiple cycles, life cycles. So it becomes a little harder sometimes to capture that activity. And so we did see some numerical dropping in the Olivo and Saltro numbers compared to that plain seed, but it wasn't quite consistent enough to split out significantly. Just like in our first year of trialing, we again saw that impact on the emergence rate. So our plain seed was slower and just had overall less emergence than both of these treatments. And we see that same trend where Saltro is a little faster, and then by that 28th day, both come together. And this year we had, again, some numerical differences in yield, about 6 bushels. And this plot yielded pretty good this year with around 48 bushels, considering how heavy the nematode pressure is in this field. So summarizing this trial up, these seed treatments are definitely aiding in the speed of emergence and the percent of emergence. And so again, these are targeted at that early planting. So those, beans that are in April, early May, that seems like it's really going to be where these seed treatments are going to be most helpful. As you move late in the season, or perhaps double crop beans where things are getting out of the ground fast, there may not be, as much of a difference there with the emergence. The Saltro treated seed had significantly less females 30 days after emergence, and the lowest numerical amount in 2020. And while we didn't see differences come out as much in 2021, a lot of those trends were moving in the same line. And we see that 3 to 6 bushel increase observed. So we had a second trial where it was a little bit larger trial. We had both those chemical treatments of Saltro and Olivo, but in this trial, we also had some biologicals, including BioST, AVO-EZ, Cloriva, and Trunemco. And so these have combination of either different, bacterial things, or in some cases, it'll actually be the dead organism where it produces a toxin. So a lot of interesting ways that we can try to approach this. And just like in that other trial, we took counts at the beginning and the end of season and had that female count. What I'm showing here is we've got the female count in this first bar. We've got the percent SCN reduction from the beginning to the end of the season, and then we've got our yield. In this plot, the Cloriva plus the base came out as our highest yield. But this is one that I wanted to be able to check on again in another year, because thanks to Jared with some of his aerial photography, we can kind of see here how it just so happened that there was this section of our field where there wasn't as much nematode pressure, and you can see that by more green. And it just so happened that the randomization, 3 of those Cloriva treatments were in that really green section. So although that one yielded the highest, we wanted to see a second year of data before we really honed in on that. And so we conducted this trial again in 2021. So you can see some of our beginning of season egg counts, end of season. And with that yield, we actually did get some spread again. That plain seed came in around 42 bushels. And this year, the Saltro treatment in this trial performed the best, and it was up at 56 bushels. That Cloriva, kind of like we were expecting, we saw some increase this year, but not quite as extreme as last year. All of the treatments were pretty well balanced within the field this year. The trial that we were running here is actually one that I'm part of in a multi-state trial. So our fields were happening here in Delaware, but there were also 12 other states and a Canadian province that were looking at this as well. And I'll give you just a little bit of some summary from the 2019-2020 seasons to bring some of this together just on a few more site years. This is a big project, and so I just have this slide here to thank everyone that I'm working with in this project, and so many different states represented, and company support and things like that. So across all of these same treatments, one of the things that we looked at here was this reproductive factor. So looking at that final egg count versus the initial egg count. So what is that reproductive factor? And on this bottom line here, you can see the non-treated about 4 in. And so many of these products were reducing that reproductive factor. And actually that Chlorella came in quite low on helping that reproductive factor to drop. This trial, we were also interested in SDS. My field did not have any SDS, but there were some of the states where SDS was present. And you can see that for SDS, index, the Chlorella plus Saltro and the Saltro were kind of there on the end performing the best. And this makes sense because Saltro is, again, a fungicide, and one of the big targets there is that it has fusarium activity. Taking a look at yield within all of these sites, that non-treated seed consistently yielded the lowest. And we can see that, again, that Chlorella plus Saltro, Saltro, and Olivo all top out at the yields. But again, if you look at this scale, we're moving from about 54.5 to 58.5. So again, kind of, being realistic, even when these seed treatments are working well, we're still kind of expecting within that 3 to 6 bushel range. One thing that's interesting to consider is that the performance of the seed treatment can also vary some on how many nematodes you have in your field. So this was broken out by fields where we had high nematode pressure, medium, and low. And when we're at that low pressure, you can get some differences that come out. And as we move into that high pressure with the blue bars, it becomes, we don't see necessarily as many differences. So knowing your nematode population through some of that survey work we're doing can also help gauge on where your success rates might be. So especially in our early plantings, we're seeing that yield advantage in the treated versus plain seed. A lot of this coming from that additional stand that's going to be out there. And then again, that nematicide piece. So having just a fungicide of any kind or seed treatment is helpful, but when we add that nematicide in, we may see that additional bushel increase. SCN egg counts can vary, and so what your population is may impact how effective your seed treatment is. And when you have SDS in your field, that's going to play a role as well. And again, for our region, this can be very spotty and inconsistent. If you have fields where you think you have SDS, feel free to contact me and let me know. Happy to come check and help see if it's SDS or other things. And again, the takeaway here, there's not really that silver bullet. We need to combine management strategies as possible. With soybean cyst nematode, that is one where corn soy rotation can be helpful. Corn is not as good of a host as soybeans, so there's some options out there. If SCN is something you're interested in, I'll just put a little bit of a plug for the SCN Coalition. This is a great website, lots of videos and resources on here if you have interest. And just in our final minutes here, wanted to briefly talk about fusarium head blight and some of our small grains research. So another one of those keywords, that first keyword was nematode, and this second keyword is fusarium. So you can jot those down. Fusarium we worry about because it gets in the head. We have these scabby kernels that are not going to yield as well, but then also have these mycotoxins that we worry about. So one of the big themes over the past couple years has been looking at fungicide timing. We've got our Feeks 10.3, that anthesis where things are actively flowering, and then the 4 days after anthesis where instead of those yellow anthers, they're now kind of a pale white or yellow. Briefly, I'll show you our 2021 wheat fungicide trial. As many of you probably, if you are a wheat grower, it was actually kind of a non-existent year for fusarium. So that was great for quality. It was an excellent quality year. There was not a lot of fusarium around. Environmental conditions were not very conducive. So in this particular trial, in the middle, we've got our FHB incidence here, and you can see it was basically 0. So these numbers that we're working with are really minuscule. That's us like out there counting really individual grains on large numbers of heads. But what we see kind of in most of our years when we have gloom blotch or SMB, we can see some activity that comes out of the Miravis products, that two-way mode of action that we don't get out of our control and things like Prosaro by itself. So I'm running a little short on time, so happy to share this with anyone afterwards if you want to look at this in a little bit more detail. One of the key takeaways that I want to bring together is that over the past couple years, there's been a lot of work across many states looking at this timing. And especially with the new product Miravis Ace having that earlier window, trying to understand when our optimal timing is. And so what we can see is that when we apply Miravis at this heading time, or that 10.3, we do get control. But when we apply at our traditional anthesis timing, we can increase that a little bit better. And then applying a little bit late also works. So especially when it comes to the dawn side of things and the mycotoxins, we see these trends again, that any of the fungicides that we're applying are going to help to lower it. But if we really target that anthesis timing in these blue bars here, you can see that's when we're going to see a lot of our biggest reduction. So those pre-anthesis treatments are effective at reducing FHB and DON, but if you have the option, timing it at that 10.5.1 is going to be where you're going to get the peak efficacy and really help to drop the DON and the fusarium symptoms. So hopefully we'll have another low year next year and won't be too much of a concern, but fusarium is one that is always back in the back of our minds from each year. So one of the things that I have a grad student that's also working on right now, this is Joe Cinderella. We currently hold the national collection of fusarium isolates where we're doing some baseline fungicide sensitivity work. And so this involves growing fungi out on all these plates where we have different levels of fungicide, and we can gauge how they're performing. And so with pitiflumethafen being a new product on the market, our goal here was just to establish baselines so that over the next decade or 20 years, we can see, are fungicides still working, or have we lost any efficacy? So these are the states that are currently involved in this project. And at the moment, everything, we don't have any resistance that's been observed. Everything is sensitive. But there's some slight differences, and you can see that our Delaware and Maryland isolates kind of range in the middle. But again, everything is working as it should, and we haven't observed any resistance, which is great news so far. We will be doing a barley survey in 2022, where we're going to be looking at some fusarium infection in fields that were planted into corn versus soybean fodder, looking at species diversity. So if you're growing barley this year and have any interest in joining the survey, please let us know, and that'll be part of Joe's graduate research project. So I'll end today with thanking everyone in my lab, our funding sources, and always thank the efforts of our great Carville research and education farm staff. I think I'm running a little short on time, but if you have questions, happy to answer now, or if you want to type into the chat, I'll be here the whole session, so feel free to drop any questions in.
All right. Thank you, Alyssa. If